The picture right in front of you, this is lilium. Okay, and this picture, this is fritillaria. Lilium and fritillaria, what are they? Okay, we are all on page number 10. Everybody must be on page number 10. You've seen two flowers right now. The two flowers which we have seen, that was lilium and fritillaria. Now these are the two flowers in which the Russian biologist, that's Navashin, it's called Sergei Gavrilovich Navashin. Okay, this is the Russian name. First was discovered, he actually discovered double fertilization. Where exactly the this question is very common for NEET also. They'll ask you who discovered double fertilization and in which species. Liliaceae family के में कौन से, so they can give you lilium and some other example, so you should not get confused. Okay, it should be lilium and fritillaria, discovered by Navashin, a Russian biologist. So let's understand what is a double fertilization. Fertilization is formation or fusion of male and the female gamete, giving rise to zygote. Zygote is a developing embryo, right? But here सिर्फ zygote नहीं बन रहा, we are also getting another product that's called as endosperm. So two things are formed, that's why we use the word double fertilization. So where it starts, let's see this. This is the common way or common process of fertilization found only in angiosperm, mostly, okay, in angiosperm. So how it starts? The starting happens with pollen pistil interaction, right? Double fertilization के सबसे पहले क्या होगा? Pollen pistil interaction होगा, मतलब anther में से pollen stigma के ऊपर गिरता है. So we have to remember all the words. So inside the stigma, that is a gynoecium. What do you remember all these things? Let's pause for a minute. You should be able to recollect the structure. ये जो structure है इसको हमने क्या पढ़ा था? Embryo sac. So अभी हम just एक quick revision करेंगे. कौन से topic का? Page number 5 के development of female gametophyte का. We are going to just revise. Just a second. This is the embryo sac. First cell undergoes meiosis, जिसके वजह से we get 4 different megaspore, right? अब उसमें से 3 disintegrate हो जाते हैं and we are left with 1. तभी हम बोलते हैं जो फीमेल गैमेट है, इट इज़ फॉर्म्ड मोनोस्पोरिक डेवलपमेंट. इट इज़ अ मोनोस्पोरिक, सिंगल मेगास्पोर से बनता है. अब होगा माइटोटिस और माइटोसिस के वजह से वी आर गोइंग टू गेट 1, देन फ्रॉम देयर वी गोइंग टू गेट डबल्ड सेल्स. So here we are just showing the nucleus part. So 2, then 4, and then 8. Out of 8, 3 migrates to the extreme chalasil end and 3 migrates to the micropyle region. So जो chalasil end होगा that would be the antipodal cells ऊपर के जो हैं, और जो micropylar region होगा उसका center वाला होगा egg cell, and दोनों के आजू बाजू we'll have synergids, right? And in the center you have 2 nucleus, we call them 2 polar nuclei, right? और secondary nucleus भी बोलते हैं. So ये था आपका embryo sac, female gametophyte का part. So this is important for you to remember. Now what is happening? Pollen has fallen on the stigma by the process of pollination. जैसे ही pollen, this is the androecium, that is the anther के ऊपर जो pollen है, it will fall. See the condition. अब आपको देख के समझ में आएगा ये कौन सा पिक्चर है. pollen का डेवलपमेंट, मेल गैमेटोफाइट का डेवलपमेंट लर्न किया था हमने. So it is A, B, C. So you will be able to recollect it is the C diagram, or it can be D also in figure 1.5. When they are falling, just a second, when they're just falling on the stigma, it is D. Okay, मतलब जो जनरेटिव सेल था वो डिवाइड नहीं हुआ था, सिर्फ वो दो न्यूक्लियस फॉर्म होता है. इट हैज़ टू बी कम्प्लीटली डिवाइडेड. इट हैपन्स इन द ग्रोइंग पॉलन ट्यूब. जैसे जैसे पॉलन ट्यूब ग्रो करता है, देन जनरेटिव सेल डिवाइड्स इनटू टू. दैट वी लर्न. देख लो आप, ओपन करो फिगर 1.5, दैट इज मेल गैमेटोफाइट. एक बार ये ओपन करो. फर्स्ट, सेकेंड एंड थर्ड डायग्राम, ए बी सी डायग्राम. उसमें से जो सी है वे यू कैन सी जनरेटिव न्यूक्लियस. दिस जनरेटिव न्यूक्लियस स्टिल डोंट डिवाइड वंस इट फॉल्स ऑन द स्टिग्मा. अब जब पॉलन ट्यूब ग्रो होता है, इनसाइड द पॉलन ट्यूब, ग्रोइंग पॉलन ट्यूब, दे डिवाइड इनटू टू मेल गैमेट्स. आपके बुक में पहले ही मेल गैमेट्स का डायग्राम दिया हुआ है, जैसे ई में दिया है. मेल गैमेट्स ऑलरेडी बन गए हैं पॉलन ट्यूब के बाद नहीं. लेकिन जनरली क्या होता है, पॉलन ट्यूब जब जनरेट होता है, दिस इज़ जस्ट एन एक्स्ट्रा नॉलेज, बट आप लोग लिखोगे एग्जाम में यही डायग्राम. यू आर गोइंग टू फॉलो दिस टेक्स्ट बुक. ओके, सो जैसे ही पॉलन ट्यूब ग्रो होता है, पॉलन ट्यूब के अंदर, पॉज फॉर अ मिनट, मूवमेंट ऑफ पॉलन ट्यूब इनटू द embryo sac, or we can say male gametes का transfer through this pollen tube is called as symphanogamy. I'm sorry, symphanogamy. This is an important word. What is symphanogamy? It is movement or transfer of male gametes, two male gametes, they are non-motile male gametes, through the pollen tube. And why are they transferred? So that fertilization can happen. So now symphanogamy is happening, and then it reaches the embryo sac. Now there is a question, how do this pollen tube know? Pollen tube को कैसे पता चलता है कि it has to follow this path? Direction कौन बताएगा pollen tube को? There is chemical released by the synergids. Okay, synergids ये egg cell है, तो उसके आजू बाजू जो है, this is egg cell, right? तो उसके जो दोनों साइड वाले कौन से हैं cells, they are called as synergids. So synergid हमने डायग्राम लर्न किया है, इसलिए इट विल बी वेरी इजी. synergid will produce chemical. That chemical will guide the pollen tube, the path it has to follow. Or for your knowledge, you should be remembering this is also called as stigmatic movement. That we'll do in the next chapter, but आप याद रखो. Then what happens? Male gametes, they are just breaking down, generative nucleus is breaking down and enters into the embryo sac. अब जैसे ही दो मेल गैमेट्स embryo sac में आते हैं, जो फर्स्ट मेल गैमेट है, it fuses with the egg cell. Okay, synergids would disappear. The second male gamete fuses with the secondary nuclei. So pause for a minute. Now let's revise. This was the embryo sac. एक्सट्रीम एंड में एंटीपोडल सेल्स थे, इस एंड में एग सेल था, एग सेल के दोनों बाजू में सिनर्जिड्स हैं, राइट? जैसे ही मेल गैमेट्स, टू मेल गैमेट्स हैव एंटर्ड थ्रू द प्रोसेस ऑफ सिम्फोनोगैमी, मतलब pollen tube के थ्रू, that enters into the embryo sac. Now what is going to happen? this male gamete fuses with egg cell and this male gamete fuses with the secondary nucleus, right? जो सेंटर में दो पोलर न्यूक्लियस थे ना, उनके साथ वो फ्यूज़ करते हैं. सो यहाँ पे और यहाँ पे दो फ्यूजन हुआ है. लेट्स टॉक अबाउट दिस फर्स्ट. दिस इज़ द फर्स्ट इम्पॉर्टेंट इवेंट जिसको हम बोलते हैं फर्टिलाइजेशन. व्हाट डू यू कॉल इट? फर्टिलाइजेशन. तो यहाँ पे क्या हुआ है? मेल गैमेट हैज़ फ्यूज्ड विद द फीमेल गैमेट. This process, this first process of fusion of male gamete with the female gamete is called as syngamy. What is it? Syngamy. Okay, this is the first fusion. Now let's see the second fusion. Second fusion में inside this embryo sac there are two nucleus, polar nuclei. उसके साथ one or the second male gamete fuses, right? तो यहाँ पे कितने न्यूक्लियस है? 3 न्यूक्लियस है. 1, 2, 3. So we call this fertilization, or this, sorry, not the word fertilization is used. This fusion, it's termed as triple fusion, right? We call it as triple fusion. So in simple word, what is double fertilization? Double fertilization का मतलब होता है triple fusion plus syngamy. Okay, syngamy कहाँ पे हुआ? Syngamy happened in the egg cell. Male gamete and female gamete fused here. Then triple fusion हुआ हमारे polar nuclei के साथ. Second male gamete has fused. So this plus this together forms double fertilization. Okay, double fertilization. So let's go to the chapter. Let's underline main points here. Double fertilization was discovered. Who discovered double fertilization? It was discovered by Navashan. In which plant? Lilium and Fritillaria. Then when does this fertilization starts? After the pollen grain reaches the pollen pistil interaction होता है. ठीक है. और फिर कौन इन्हें गाइड करता है? pollen tube जैसे ही ग्रो कर रहा है, who is guiding it? This is an important thing you should be knowing. It is guided by the, see, let's read this. The growth of pollen tube is guided by the chemical secreted by synergid. It's an important question. Okay, so many questions are there. अब guide करते वक्त there are 3 ways how it can happen. 1, through the micropore, micropylar region से जब entry करते we call them porogamy. This has already been covered. No diagram हमने दिखाया है 1.12 में. Then if it is coming through the chalazel region, chalazogamy बोलते हैं. Or if it is coming through the integument region, this diagram we have already discussed, so I'm not just wasting time in that. ठीक है, you have to remember the movement or entry of pollen tube into the ovule. 3 types depending upon the entry position. Okay, one is the micropyle, then another is chalazel, and the third one is the integument region. Then important thing that you have to remember is सिफिनोगैमी. सिफिनोगैमी का मतलब है नॉन मोटाइल मेल गैमेट्स को pollen tube कैरी करती है. और what is the purpose? To ensure fertilization. That will give 100% fertilization. Then what do you mean by double fertilization? Double fertilization is syngamy and triple fusion together that happens. Okay, now what is syngamy? Syngamy is fusion of haploid male gamete with haploid female gamete. So though haploid, they are coming together, they form diploid zygote. So this is the first advantage of double fertilization. We get haploid comes together forming diploid zygote. Now the second important fusion that happens here, it's called as triple fusion. Triple fusion में क्या होता है? Second haploid, or we can say second male gamete, fuses with the secondary nucleus, producing PEN, primary endosperm nucleus. Endosperm means it's the food. Okay, पहला बना हमारा zygote, अब उस zygote के लिए food भी बना, मतलब it's the nourishing tissue. Okay, primary endosperm nucleus is nothing but the nourishing tissue, and we get triploid endosperm क्योंकि उसके अंदर 3 न्यूक्लियस होते हैं ना, so that's why we say it is the triploid endosperm. But what is the word given for it? In the center, when you are drawing this diagram, no, when exam mein it is asked draw diagram of double fertilization, you have to draw this diagram showing triple fusion and syngamy. It has to be clearly shown. Okay, so make sure you're drawing the diagram of double fertilization and explain what happens in double fertilization. Okay, 2 types of fusion is happening, both have to be explained, and the diagram is important.